All right, so up to 36, I spent talking about plasma membrane. Now, the exocytosis, endocytosis, I'm going to mention these real quick. Our cells, because they are plasma membrane, okay, they have the ability to wrap around a substance and bring it in, and their materials that they make, they can actually spit them out for exocytosis, which I think is kind of cool, okay? So they deserve a mention. So now, as we move away from the plasma membrane, cytoplasm. This is that inner portion that contains a lot of those organelles. The cytosol is going to have like the materials that the cell can use, okay? And now, cytoskeleton helps to give shape, but I will say for the side, I'm trying to get to my nucleus. I give it one slide. Compared to how many for the plasma membrane? 36. So, all right, plasma membrane. All right. Now, that plasma membrane, being that selectively permeable membrane, it is really helping that cell survive. Now, I'm mentioning the cytoskeleton because here's what has to happen for animal cells. Because we are just a plasma membrane, the cytoskeleton is going to allow this animal cell to be able to do this number. For some of the cells of our body to be able to flatten and poof back out. We have areas in the body that do that. Okay, it is really cool. All right. So the cytoskeleton for the structure allows for this cell, this animal cell, to be fluid in its movement. As we look at the inside of the cell, as we move from this outer portion, moving inward, getting to the nucleus and other organelles, the one thing that I find very interesting about this is how, with the cell having a lipid bilayer, these organelles do too. The nucleus has it. The nucleolus has it. Endoplasmic reticulum has it. The Golgi apparatus has it. And there's a reason why. Every one of those plays a part in the product that that cell makes. And that product either stays in the cell or it travels to the plasma membrane. And in doing so, it gets wrapped in a double membrane. So that if it has to go out of the cell, that it gets moved to the plasma membrane where it either just kind of pushes out or spits it out. That's pretty cool. And it's in a bilayer membrane. So, the nucleus, with our nucleus, it has a double membrane. It has the bilayer. It contains nucleoplasm, our nucleolus, nuclear envelope, and the DNA is located in the nucleus. Ta-da! It's safe. The DNA is safe right there. That DNA is never going to leave the nucleus. It will be RNA that will leave, and that's where problems can come in. Things can damage DNA, such as UV light, okay? But that doesn't mean it got out. It's damaged through the cell. Our chromosomes, when they are in the cell, when they're in the nucleus, they exist as chromatin until it's time for the cell to make a copy of itself. The DNA does not become a chromosome until prophase occurs. Well, interphase, until we begin cell division. So it remains as chromatin in this form that it's existing. It's allowing for easy access to a gene. So if this cell, okay, if that's my nucleus and this is my cell, if this cell is responsible for making something, okay, the gene needed will remain pretty exposed until it's time for that cell to duplicate. Does that make sense? So it exists as chromatin. When it comes time for the cell to make a copy of itself, it'll condense. We'll get, because it makes a copy, they come together into the chromatids. We think of them as the chromosomes. They're held together by the centromere, and then they begin to go through cell division. Our ribosomes, this is our site of protein synthesis. We're going to talk a little more about ribosomes in Chapter 4, but they have a large and a small unit. We have ribosomes that are free, meaning they do their job in the cytoplasm of the cell. If a cell has to make a product to leave the cell, we need the endoplasmic reticulum. That's the difference. Free ribosomes make something that stays in the cell. Endoplasmic reticulum makes products that leave the cell. That's the major difference in the ribosomes. Their job, take that messenger RNA strand, use the code, and make a protein. Their job for the ribosomes is to help string together the amino acids. It's putting together the primary structure of the protein. That's what the ribosomes are going to do, which is pretty cool. Take a break. Be back at 3:30.